last but not least. It's my pleasure to introduce our next speaker, James Barlow, President and CEO of ImmunoGenesis. James is leading one of the most exciting clinical stage immuno-oncology companies in the field today. Under his leadership, ImmunoGenesis is tackling one of the biggest challenges in cancer treatment, which is immune-excluded tumors. These so-called cold tumors that don't respond to current immunotherapies like PD-1 inhibitors. The company's leading candidate is IMGS001. It's a next-generation dual-specific PD-L1, PD-L2 antibody with a built-in cytotoxic function designed to break through the stromal barriers that prevent immune cells from reaching tumors. In early signs of doing what no other PD-1 inhibitor has been able to do, which is reprogramming the tumor microenvironment and delivering clinical benefit in patients who've already failed checkpoint therapies. ImmunoGenesis is also advancing IMGS101, a hypoxia-reversing agent that may make otherwise resistant tumors like pancreatic and prostate cancers responsive to immunotherapies. With over 60 million raised to date, strategic support from institutions like MD Anderson and the Cancer Focus Fund, and an impressive early data readout, ImmunoGenesis is well positioned to lead the next wave in immuno-oncology innovation. Please welcome James Barlow.
Thank you so much for that introduction. Excited to be here and share the ImmunoGenesis story with you. So we have a really small goal at ImmunoGenesis. We're trying to show that we have a better version of Keytruda, the $30 billion monster that's the best-selling drug of all time. But we really believe that we're going to drive the next turn of innovation in oncology. Our founder was one of the trailblazers of the original checkpoint inhibitors, and we really set out to design and overcome a lot of the barriers and challenges that have faced the first generation. So what is the problem that we're trying to solve? So we really believe that you need a superior PD-1 pathway drug to drive the next big transformation in immuno-oncology. Why do I think that? Well, Keytruda is a great drug, but its efficacy is quite limited. It works really well in melanoma and lung. It has a number of other approvals, but the efficacy is quite modest. And when you look at all cancer patients, only about 15% derive a clinical benefit from a PD-1. And what the industry's tried to do is they said, okay, well, let's just throw combinations at this. We have this amazing drug. If we throw combinations at it, that's bound to work. The problem with that, and actually Merck is one of the biggest proponents of this, is if each drug doesn't have underlying monotherapy efficacy, when you put them together, you're not going to get synergy. When you have 5% and 5% response rate, you put them together, you get 10. You don't get 30. And actually, Merck, their external collaborations lead, sits on a lot of panels with our founder, and he'll say to the audience, don't even come to us to partner with Keytruda if your drug doesn't have monotherapy efficacy, because if it doesn't work as monotherapy, it's not going to work in combination. And I always want to jump out of my seat and say, yeah, but isn't the problem that Keytruda doesn't have monotherapy efficacy in 80% of cancers? And so what we've seen is 1 out of every 287 combination trials of immuno-oncology has been successful. And I don't need to be a mathematician to say that that's a rather dismal success rate. So we really believe that our drug, the way we've designed it, can drive this transformation because it has a multitasking mechanism. It does 2 things. It blocks the PD-1 inhibitory signal, but it also can kill cells that express PD-L1 and PD-L2, which form the wall around tumors. And I'll show you more information on the MOA as well as our early data. So high level on the company, IMGS001 is our PD-L1, PD-L2 dual-specific inhibitor. It's been engineered for first-in-class efficacy in immune-excluded tumors and best-in-class efficacy in tumors where the PD-1s have already shown to be effective. As Eric mentioned, we also have a second clinical asset, IMGS101, that reverses another immune barrier, which is the barrier of hypoxia. T cells won't traffic to hypoxic regions. This drug reverses that hypoxia, allows the T cells to traffic, and then you can give a checkpoint inhibitor and have it be effective. Our founder, Mike Curran from MD Anderson, he worked closely with Jim Allison on the seminal discoveries that were made that led to the Nobel Prize. And Mike has had his own lab at MD Anderson for the last 13 years, really focused on how we overcome the immunosuppressive barriers to expand the use of immunotherapies beyond melanoma and lung. We've raised about $70 million since founding. That includes $15.5 million from CPRIT, through the government of Texas, through the state of Texas, as well as $4.5 million from the Cancer Focus Fund, which has been a tremendous supporter for us. And then we've raised about $50 million, mainly through high net worth and family offices, which is going to drive us to a data inflection point, and we believe allow us then to raise an institutional round later this year, which will drive us to proof of concept data. And what's exciting is when we look at the landscape, this drug would come to market and be approved simultaneous to when Keytruda and Opdivo are losing exclusivity in 2028, 2029. So there's real potential for this drug to become the new foundational agent with IP protection out to 2042. So just taking a look here, this is difficult to read, but I'll just hit some of the highlights. So we've made a lot of progress over the last 5 years. We've, as I mentioned, raised about $70 million, brought 2 assets to the clinic, built a world-class team, and we're really excited about how we're positioned to show proof of concept and drive the next big value iteration for the company. Some of our key investors include Cancer Focus Fund, Addit Ventures, as well as BioAdvance and GenHen. And then we have some key partners, including, I mentioned CPRIT, and we just opened our site here at Oxnard, and we're really excited for that. We think they're going to be tremendous supporters of the program and be able to rapidly enroll and really be a linchpin to the success in being able to show the initial proof of concept. So let's talk a little bit more about IMGS-001. So we know that the PD-1s and PD-L1 drugs have been a major success. It's estimated that they're going to do $50 billion in sales this year. Keytruda alone may do 30. It's the best-selling drug of all time. But the reality is they're only relevant for about 45% of tumors, and there's a tremendous unmet need in the immune-excluded tumors, which represent a majority of cancers. Underscoring this a little further, if you look at all cancer patients, about 45% are eligible via the indications for a PD-1 or PD-L1, but only about 15% actually get a clinical response. And so you can see that there is tremendous upside to improve on these response rates. So this gives you a sense of where we're targeting. So if you look on the left of this slide, the immune inflamed, this is where so these are slices of tumor. The brown spots are T cells. And in the immune inflamed, this is where the PD-1s have been very successful. You have a lot of your T cells that are right around the tumor. If you block the PD-1 signal, those T cells are activated, they can kill tumor. However, most cancers sit in this middle bucket of immune excluded, where you have T cells in brown, but they can't get to the tumor. And so you can block all the PD-1 signaling you want. If the T cells can't get where they need to go, they're not going to be effective. The analogy I like to use is if you have a bunch of soldiers attacking a castle, if those soldiers are right at the castle gates, if you can give something like Keytruda to open the gates, they can rush in and be effective. But most tumors also have a castle wall. And if the soldiers are stuck outside the wall, you can open the gate all day long. It doesn't matter if they can't get through the wall. And so our drug's been designed to both take down the wall and open the gate. And what does that mean from an opportunity perspective? Well, if you look on the right, this is the distribution of these phenotypes by the various tumors. And so on the bottom is colorectal cancer. Very little colorectal cancer is inflamed. That little red area you see there, that's actually MSI high, which is only about 5% of colorectal. 95% is microsatellite stable. And the PD-1s have basically been a 0 in microsatellite stable. They've shown no benefit. 65% in blue of colorectal is immune excluded. So our drug has been specifically designed to deal with exactly that scenario. So we think in colorectal we could be a 30+% response rate because of our mechanism. But even in lung, where the PD-1s have been successful, if you look at lung, the red bar there, immune inflamed, is about 30%. Lo and behold, the response rate for PD-1s is right about 30%. We're going to do just as well in that 30%, probably a little better. But another 45% is immune excluded. We're going to do significantly better in the blue bar. So we think in lung we could be a 50 to 60% type response rate as a monotherapy, and then add on to that as with any type of combination. So when you look at this distribution of these different immune phenotypes, we believe we're well positioned to not only show proof of concept in things like colorectal and ovarian where the PD-1s haven't worked, but also improve in the tumors they have, such as lung cancer. So this is what it looks like when I talked about immune excluded. So essentially you have your tumor in blue. Three cell types are really critical to creating this barrier. So your MDSCs, cancer-associated fibroblasts, and tumor-associated macrophages create a wall that keeps your T cells out. And as I mentioned, the industry said, okay, let's try combinations. And so there's been thousands of combination trials done either through bispecifics or through PD-1-based combinations. And what you've seen is 1 out of every 287 be successful. And so we believe you need a different approach. So imagine that you had a single drug that could both take down that immunosuppressive wall and has best-in-class blockade of the PD-1 signal. So that's exactly what IMGS001 has been designed to do. It can kill the cells that create the wall, and it also has best-in-class activation of those T cells once they get into the tumor. And here's what the design of the molecule looks like to make that happen. So it's actually a single antibody. It's not a bispecific. It targets PD-L1 and PD-L2. There's about a 30% conserved region between those two ligands. And so we took advantage of that in designing this molecule where both arms bind with strong affinity to both ligands. And that's really important because the drug is bivalent and works just as well in a PD-L1 dominant environment, a PD-L2 dominant environment, or in a situation where you have both ligands present. It also gives us a lot of optionality down the road. If we want to add a third target here, we could swap out one of the arms and we could add in, say, VEGF. And at that point, we would have a true bispecific that's hitting 3 important targets. From a blockade perspective, if you look at the lower left, we're essentially the best of both worlds between a PD-1 and a PD-L1. We block all 3 important inhibitory interactions, whereas the other drugs just block 2 of the 3. This is an incremental advantage. The real step change is the killing function that's built in. So you see on the FC region here, we've engineered 3 different mutations to drive robust ADCC and ADCP. And this is a perfect example of how we've learned from the challenges in the industry. Others have had this vision. Pfizer really wanted their PD-L1 avelumab to have the ability to kill, but they use an unmodified IgG1. And the reality is when you use an unmodified IgG1, you need a lot of NK cells in the microenvironment to drive that cell killing. And you just don't have that in solid tumors. Our founder was on the SAB. He saw that firsthand, and he said, if I'm going to design a molecule that actually has robust killing, I'm going to need to use a different approach. And so we've engineered in these 3 different mutations. They're actually were originally designed by Xencor. And these 3 mutations drive 500 to 1,000-fold greater cell killing versus an unmodified IgG1. And they do that by relying on other cells such as MDSCs and macrophages and neutrophils that are plentiful in the solid tumor microenvironment. So this is a perfect example of where science is hard, and we've learned from the last 10 to 15 years of challenges, and we've engineered in these mutations that we believe are going to drive really robust cell killing in patients. So here's what the clinical trial looks like. It's a phase 1A, 1B. The 1A is a traditional dose escalation. We have 5 different dose cohorts. We'll enroll up to 25 patients across solid tumors. We'll look at safety and early signs of efficacy in this patient population. And then by about July of this year, we're going to expand into our 1B. In our 1B, this is the tumor-specific cohorts. And what you see there is we have 2 groups. Colorectal and ovarian are examples where you don't have any PD-1s or PD-L1s approved. So IMGS001 would be the first PD-1 pathway drug in those patients. But in bladder, triple-negative breast, and gastric, you do have PD-1s and PD-L1s approved, but the efficacy is quite modest. So our drug would come in for patients who are refractory to those drugs, and we would attempt to show that we can reverse that resistance. We're going to look at biomarkers in the 1B, look at PD-L1 greater than 5%. The reason we want to do this is we want to select for that immune-excluded group. As a monotherapy, we can't deal with immune desert where there's no T cells there. We know they're not inflamed because they're either refractory to PD-1s or they're in tumors where PD-1s don't work. But we want to make sure that they're in that immune-excluded subtype, which is why we're selecting for PD-L1 greater than 5%. So let's get into some of the fun stuff. What does the data look like so far? So based on our preclinical modeling, we didn't think we'd see any efficacy until we got at least to 5 mg per kg. In mice, you had to be at least at 5. Based on some of the pharmacokinetics, we thought you had to be at least at 3. And if you look between the lowest dose of 0.3 and the highest of 15, there's about a 75-fold delta in terms of exposure. But what we've actually seen is really exciting clinical benefit at these lower doses. So the first patient in the trial was a colorectal cancer patient, so it was right across the plate for where we're trying to show differentiation. This patient had rapidly progressed on 3 other treatments, including 2 chemotherapies and an experimental PD-1. His CEA, which is a key blood marker for colorectal, had been going up like a hockey stick. He came on IMGS-001. We leveled off the CEA and then brought it down by 30%. And this patient had 6 months stable disease, and he was getting 1/30th of our target dose. I really believe that if he was able to get a higher dose, we would have saw a response because he did so well at such a low dose. So then we moved up to the 1 mg per kg, and those first 2 patients you see, the first 2 blue lines at the top, those patients are still on drug. So the first patient had progressed on 2 different chemotherapies as well as Keytruda. Came on IMGS-001. She'll be on drug for a year as of April 26th, doing phenomenally well. Nasopharyngeal cancer that was metastatic to the lymph node and liver. Her tumors overall are down about 23%, and one of her liver tumors is down 81.5%. And she's doing tremendous clinically. That's what the scan you see on the slide here is actually of the liver tumors for that patient. And then we have a second nasopharyngeal patient who's now been on drug over 8 months and also seen tumor regression. And this patient had actually failed 3 different PD-1 inhibitor regimens. So what we're seeing early on is some clear differentiation versus the existing PD-1s. First, we're showing efficacy in patients or we're showing clinical benefit, I should say. It's not quite responses yet, but we're seeing clinical benefit in patients who have progressed on PD-1s. We're also seeing this deepening of response over time. Typically with the PD-1 drugs, you get your best response at the first or second scan, and you don't tend to see further tumor regression. We're seeing, if you look at the first nasopharyngeal patient, every scan has been more and more tumor regression over time. And one of the third key areas is we're seeing impact on liver tumors. The current PD-1 drugs typically don't impact the liver tumors, and so the fact that we're seeing this significant shrinkage in liver tumors is a really exciting sign. It's early on, it's a small number of patients, but I think what's important is that at these low doses, we don't believe that we're saturating PD-1 blockade. So what we're seeing here is actually being driven by the killing function of the molecule, which is completely unique. And so once we get up to 10 mg per kg, which is where we're dosing right now, both arms of the mechanism are going to come into play. So we're excited to see what we can do in those patients. And I think what's exciting here too is these you don't expect to see a lot in your first group of patients. These are very sick patients, end stage, 4th or 5th line, and so to see patients come on at very low doses and do quite well is an exciting sign of things to come. So just to put a bow on IMGS-001, so we really believe it's going to transform this space. It's been engineered, as we talked about, to address this issue of T-cell lockout. And we think it could outperform Keytruda really across the full spectrum of cancers. But we're attempting to show proof of concept first in the immune-excluded subtypes where the PD-1s haven't worked. It can then become the superior foundation for future combinations. And as I mentioned earlier, it would get approval simultaneous to when Keytruda and Opdivo are coming off of exclusivity. So really exciting opportunity here to have the next foundational drug in immuno-oncology. Before I go on, yeah. Yep. So we haven't, none of the patients so far have, it's a great question and really important. None of the patients so far have been selected on that basis. Right now it's an unselected population in the dose escalation. In the tumor-specific cohorts, the reason we did that is we're the first company to target PDL2. And so it's unclear what level of PDL2 expression is important. And the other piece is as a monotherapy, we can't deal with the immune desert patients. So by using PDL1 greater than 5, you gain two things there. One, you have a sense that you have an existing immune response. So you are selecting for that immune excluded population. And then the second piece, when you have PDL1 greater than 5%, there's an extremely high correlation with PDL2 expression. So it's another way to have a proxy that patients are also expressing PDL2. However, I mean, as we're looking at this, the patient who's doing the best and is almost out a year, she was PDL1 negative at the tumor. And so it is something that we've been having some conversations, and we're going to talk to our clinical ad board about how we think about that and whether we reconsider that, because there is an example here of somebody doing extremely well. Because our drug's not only targeting the tumor, it's targeting the microenvironment. So you might miss some patients who have L1 and L2 in the microenvironment if you just look at the 5%. Yeah, it's a good question. I mean, it's still a significant percentage. I mean, I think you're still in the 50, 60% of the tumors. A lot of people think, like colorectal, it hasn't worked, so it must be cold. But actually, there's a good amount of colorectal. Like in the slide earlier, 60% are considered immune excluded. So those would be examples where they do have an immune response. So a lot of times, it's not that they're cold, it's that they have these immune barriers that are causing the lockout of T cells. So actually, our next product is we're doing in pancreatic. So that's a perfect lead in to our next one. But before we do that, is there another question on 001?
Yeah, just along the lines of characterizing tumors. Do you have MSI data on any?
We know that the colorectal patient was a microsatellite stable, and the other ones, I think we do have that status. I don't think any of them have been MSI high to date. That's a good question. So if we look at our second drug, IMGS 101, this drug has the unique capability to reverse hypoxia. And so if we look at certain tumors like pancreatic, head and neck, and prostate, 50 to 60% of the tumor is hypoxic. And T cells won't traffic into those hypoxic zones. And so how do you expect to give a checkpoint inhibitor and have it be effective if their substrate's not present in the majority of the tumor? And yet Merck and BMS spent billions trying to show their checkpoints could be effective and to no avail. And so this is, again, where science is hard. And our founder said, unless you deal with the hypoxia, there's no way checkpoints are going to work here. And so he found this drug that uniquely reverses hypoxia. It's actually a hypoxia-activated prodrug. But actually, after you give about 2 doses of it, the hypoxia goes away. And so what you see is by reversing that, the T cells can traffic in, and now you can give a checkpoint inhibitor and have it be effective. And so there was a phase 1 done at MD Anderson where they looked at combining this drug with the CTLA-4 inhibitor, ipilimumab. And the efficacy was quite striking. So this was a diffuse trial, 4 different doses, 4 different indications. You don't expect to see a lot. The patients, pancreatic, they had to fail 2 lines of chemo. So they had about a 12-week estimated survival. Head and neck, they had to fail a PD-1 and chemo. And prostate, they had to fail a hormone and chemo. And despite that, we still saw a 17% response rate and about an 83% disease control rate. If you look at the spider plot in the lower right, the 2 light orange patients in the middle, those are actually pancreatic patients with stable disease out past a year. So these were patients that were end of life and had significant PFS. And then there's a prostate patient on the bottom that's a radiographic complete response. This patient had 15% of their liver was tumor and had failed 2 lines of chemo and 2 lines of hormone and had a radiographic complete response with this combination. Patients were doing so well that 5 of them came back and asked for retreatment because we only treated in the first 12 weeks. And so there was an amendment put in, and 4 of the 5 had further regression after retreatment. So we believe in this trial that we undertreated and that having a PD-1 on board as well would have significantly enhanced the efficacy. And so in the phase 2, we're actually adding a PD-1 as well as CTLA-4 with the hypoxia reversal drug. So we're using the 2 drugs from Agenus. Zalifrelimab is their CTLA-4, and balstilimab is their anti-PD-1. The reason we selected these is they have about a 40% grade 3 rate, whereas Ipi-Nivo has about a 60% grade 3 rate. So having a triple combination, you certainly got to look at toxicity. So we're going to look at these drugs across pancreatic, head and neck, and prostate. And prostate, there's a built-in Simon two-stage design, as we already have a large pharma partner who's quite interested in this drug for prostate cancer. So in my last couple minutes, in summary, we are raising a Series A prime financing right now. We closed our Series A in September, but because this data looked quite promising, the early data for 001, insiders wanted to put more money to work. So we opened up an A prime round. We've raised about $9 million in the A prime round. We're trying to get to 12 to 15 in that round. That closes at the end of April. That gives us runway into the first quarter of next year. And we expect to have value inflection data around September. So around September, we'll have data in 25 patients for 001, the PD-L1-L2, and we'll have data in about 10 patients with the triple combo for 101. And we think based on that data, we can raise our Series B, and the Series B can drive to proof of concept. Because by the end of 26, we should have 80 patients of data with the lead and about 30 patients of data with 101. And for our Series B, we're thinking of right now of a $75 to $100 million type raise that would allow us to complete both of these trials, as well as look at the expansion where we see promise to be able to rapidly expand the cohorts for IMGS 001 as we move forward into 2026. We have a great team. We're lean and mean. We have 10 FTEs. Our group has a lot of expertise, not only in their subject matter, but a lot of them have been executives or CEOs at other biotechs, and that's really helpful as we have laid out our strategy, laid out our regulatory approach, and so the team has done a tremendous job, and we'll be adding to that this year as we raise the Series B. So in conclusion, we believe that we have transformational agents that are going to drive the next big advancement in immuno-oncology. In particular, 001 has the chance to be a foundational agent in a $100 billion market. We also are excited about the phase 2 for 101, and we already have pharma interest there. And we think by the end of 26, we could have proof of concept data that could potentially lead to an exit. So let me stop there. There's a few minutes left if there's any other questions.